For years, we've been told that DNA is the key to our health. But what if I told you the real story is happening right now, in real time, inside your body? Today, we're diving into how AI is finally letting us read that story by unlocking the secrets of our proteins. So let's start with a really big question. What if we can know exactly what's going on with our health at any given moment, all from a single drop of blood? You see, our genes, our DNA, that's just the blueprint. The actual story, the story of our health as it unfolds, is written by the tiny machines that bring that blueprint to life, our proteins. So to really, truly understand our health, we've got to look beyond the static DNA code. We need to step into the incredibly dynamic, ever-changing world of the proteome. That's the complete set of proteins hard at work in our bodies right this second.
Right. We've all heard the analogy a million times. DNA is the blueprint for life. It's got all the instructions, all the potential. But think about it. A blueprint can't build anything. It just sits there. So who's actually doing the work? Who's building the house?
This slide really nails the difference. On one side, you have genomics, studying our genes. It tells us what could happen, our risks and predispositions. But on the other side, you've got proteomics. That's the study of our proteins, and it tells us what is happening. They're the active workforce, showing us the biological reality of our health in real time.
And this workforce, it's just mind-bogglingly complex. We have around 20,000 genes, but they don't just make 20,000 proteins. Oh no. Through all these complicated processes like alternative splicing and post-translational modifications, our bodies create millions of different protein forms, each one with a highly specific job to do.
Okay, so if these proteins are telling this constant dynamic story about our health, how in the world do we listen in? How do we measure these invisible workers? Well, it turns out you need a machine of almost unbelievable precision.
And here it is, the mass spectrometer. The easiest way to think about it is as a hypersensitive molecular scale. It is so ridiculously precise that it can weigh the individual building blocks of proteins, which lets scientists figure out exactly what they are.
So let's follow a protein on its little journey through this machine. First, you take a sample, like blood, and the proteins in it are chopped up into smaller pieces called peptides. This super complex soup of peptides gets separated and then shot into the spectrometer. The machine weighs the whole peptides, then it literally smashes them into fragments and weighs the pieces. It's this two-step weighing process that gives each peptide its unique fingerprint.
Now, what comes out the other end is just an absolute flood of data. A single sample from one run can generate more than 1 million distinct signals. This isn't just a simple measurement. It is a gigantic computational puzzle that needs to be solved.
And that massive data puzzle brings us to our next huge challenge. How do you make sense of it all? It's kind of like trying to hear a single whisper during a Category 5 hurricane. How do you possibly find the real signal in all of that noise?
Okay, so an experiment kicks off with just a chaotic mess of about 20 million raw data points, or what they call spectral peaks. That's the hurricane of noise we have to sift through.
So the first step is to bring in some really sophisticated algorithms. They connect all these points across time, filtering out the random noise. And just like that, the hurricane is calmed down to about 1.5 million potential signals that just might, be actual peptides.
And for the final step, this much cleaner, much more manageable data set is checked against a huge database of known proteins. And that data storm, that 20 million points, gets funneled all the way down to, say, 5,000 proteins that we can confidently identify. It's pretty incredible.
Now, for a long time, that final step of matching the data to the database was a massive bottleneck. We're talking hours and hours of computer time. But now, artificial intelligence has completely changed the game.
Just to give you a sense of the scale here, it's staggering. To identify all the proteins in just one sample, a computer has to make literally billions of comparisons. It has to check the data it collected against every single potential peptide in its database. It's a colossal task.
And this is where things get absolutely wild. AI-powered tools have taken that monumental task, that computational marathon, and they've turned it into a 100-yard dash. A search that used to tie up a computer for 10 hours can now be done in 9 seconds. No, that is not a typo. 9 seconds.
But here's the thing. It isn't just about raw speed. It's about being better. Because AI can look at dozens of different data features at once, It actually improves the number of proteins we can confidently identify by more than 14%. So you get more speed and more accuracy. It's a total win-win.
So, this incredible new power isn't just some cool academic exercise. It is unlocking real, tangible applications in medicine, in the clinic, that can help people right now.
And the proof is already out there. For diseases like COVID-19, looking at protein panels can predict how sick a person will get way better than standard tests. For liver disease, it can mean replacing painful, invasive biopsies. It's happening across the board. Proteomics is already starting to outperform our existing clinical tools.
The big takeaway here is that this isn't some experimental science anymore. It's mature. Researchers now have a whole toolkit of different methods. You can see some of their technical names here, but what's important is that they can now pick the perfect tool for the job, whether they need to analyze thousands of samples very quickly or get pinpoint accuracy on just one. And all of this is building towards one ultimate goal, kind of the holy grail of this whole field, using the proteins in our blood as a liquid biopsy. Just imagine a simple, routine blood draw that gives your doctor a complete, real-time snapshot of your health, catching problems long before you ever even feel a single symptom.
And that leaves us with this final, really provocative thought. By truly learning to listen to the dynamic, real-time stories our proteins are telling us, we are moving so fast towards a future where we might not just treat diseases better, we might actually be able to predict and prevent them from ever happening in the first place.